I'm really very excited to be here to have the opportunity to share with you some of the stuff we're doing in my lab right now. But first, I have to thank these girls. They are my students. They are really the reason why I'm here. They are the ones running the lab. They are the ones that do all the experiments. They are the ones that do everything. I'm almost like useless person in my lab. So they are the reason why I'm here and why I have like all these amazing results I'm going to share with you. So, I've always been interested in understanding how the evolutionary process is in plants and insects. So I mainly focus on the evolution of plant defenses, particularly resistance and tolerance to herbivory, and how these defenses exert evolutionary selection processes in life history traits in the insects. For example, survival, consumption, and how again these changes in the life history traits also again changes the evolution of defenses. And so when I started my own group, I decided that my lab was really easy and I wanted to complicate a little bit more. And so now I wanted to know how all the bacteria that are associated with the insects and with the plants are shaping this evolutionary process. And so one of the big questions we're trying to answer in my lab is essentially this, if we can understand better plant-insect coevolution by studying how the effects of the bacteria associated with plants and with the insects. So this is my study system. So the plant is it's a rural plant, it's natural, it's really toxic, and this is the beetle, it's a specialist beetle. It's called Lemma daturacula, it really doesn't matter. And because I wanted to understand the evolution of the herbivory interaction, we went to the field, and so we collected tissue, like seed endophytes, foliar epiphytes, those are the ones that live on the surface of the leaf, foliar endophytes, and for the insect we also collected eggs, the whole gut of these little larvae, and its frass. And we started by doing what everybody was doing. So we took the tissues to the lab, we extracted DNA, we did the sequencing analysis, and Sienna was the one that started analyzing the bacteriomes. And so we started doing the description of the abundances and then the diversities for the different tissues we have for the insects and the different tissues we have for the plants. And we also were interested in understanding if there's a core bacterium of the interaction, not only for the plants and the insects, but a bacterium that can help us understand the interaction. So we found, you cannot see here, 85 amplicons that are shared among all the tissues. And we also did some functional inference, and so again, these are the tissues from the insect, the tissues from the plant, and we found some microorganisms that are involved in the degradation of toxic elements, some others that are involved in the defense mechanisms of the plants, and also some that are involved in the detoxification mechanisms. And this was really interesting, but the ecologist in me felt that we were doing nothing really novel enough to explain the interaction, because we were doing what everybody was doing, but just for the plants and for the insects. And so what we decided to do was to start building something that it's called the multi-layer network approach. And so with the greatest collaborator of all, he is my husband, right? So Roberto told me we can start building multi-layer networks. And so what we have here is coabundance networks. And so for example, for the insect, each node is the bacterial group. The size of the ball represents the abundance. So if you see a really tiny ball, it's because this bacterial group is really not abundant. And then we have some others that are really abundant. And if you see that, you cannot see here, if those two nodes are linked, it's because there's either a positive or a negative correlation among the abundances of those two bacterial groups. And so here for the insect, you can see that some nodes have really a lot of connections with some other nodes, and some of them are have less links among them. And we can do the same thing with the plant. And so, because obviously when the larva is eating the leaf, it's eating everything that is on the surface of the leaf and inside the leaf. So we have the same bacterial groups on both organisms, right? But the structure of the network is different. So here we can see that although these two nodes are in both organisms, like the abundance is different, and the connections among them is also different. So the structure of the community is really different. And now that you have like the network, you can start studying like different properties of those networks. So for example, there's something we call centrality, which is essentially like there are some nodes that have many links and some other nodes that have only like 1 or 2 links. And so here, for example, in the insect, we can see that there are some nodes that are really linked among them, but they lost their connections like with other nodes that are present in the plant, right? So the structure and the importance of each bacterial group is, although they are shared among both organisms, they are really different. And so they tell you like something that is not about the presence or absence of certain bacterial groups, but how they correlate in their abundances that may help us to understand plant-insect interaction. And so this is really this is really amazing. I'm really happy about this network. But again, the ecologist in me said, so this is the network of co-abundances. And even though you find like a positive correlation between this group is too abundant and this other group is also very abundant, it tells you nothing about the ecological interaction, because it's not a necessary result that even though they are both super abundant, it's not necessarily because they relate ecologically, right? So somehow I was able to trick one of my students into doing like all the possible experiments to actually calculate the ecological interactions among all the groups we have. So we were able, Anna was able to isolate 9 different species of bacteria from the surface of the leaf and 8 different species of bacteria in the gut of the herbivore. And so what we did was something that it's called a spots on lawn experiments. So basically you have like a petri dish, and on the lawn of the dish you grow 1 bacteria, and then in each one of the spots you put the other bacteria. And so we have controls because we want to know like the growth rate of a certain bacteria without interaction. And then, for example, we have one set of experiment that is on the lawn of this petri dish. We grow 1 bacteria that comes from the gut of the insect, and in each one of these dots it's like a bacteria that comes from the leaf, and we have the other effect. And so with this formula, we were able to estimate like the effect of the interaction. So this is essentially like a difference in growth between when each bacteria is growing without the interaction versus how it does when it's interacting with another bacteria. And so this tells you like the effect of the interaction. And now with this, we can build a matrix that tells you like all the possible ecological interactions among all the possible combinations between bacteria from the plant and from the insect. And with this, we were able to actually build something that you cannot see. But this is the network. Again, it's a multi-layered network. So these are the nodes from the bacteria of the surface of the leaf, and these are the ones from the insect. So this is like the real ecological interactions that we can see among those bacteria from these 2 different organisms. And I know you cannot see, but here we found some interactions that are actually positive interactions, but those you can only find them among gut bacteria associated. And mainly what you can see is that everything is red. So that means that most of the interactions we see, there are actually antagonistic interactions. And so these are like bacteria-bacteria interactions, right? So we were next like really interested if these interactions that you see in a petri dish among the bacteria you isolated from the plants and the insects can actually affect the insect. So Sanya did this experiment. We went to the field, we collected some leaves, and at the same time we collected 2 leaves from the same plant. And one of the leaves was treated with a serial of washes so that you can actually remove all the microorganisms that are on the surface of the leaf. and the other leaf was like the control one. So we have one leaf that has everything on top of it, and the other one that is surface sterilized. Right? And we place these leaves on petri dishes, and I mean, I can tell you that there's a little larva here, but I think there's nothing here. But so we place a tiny larva here, and we measure like survival, mass, consumption, efficiency, like how long it spends to complete its development at adult mass. And to be honest, I wasn't expecting anything. Like I thought, like there's no way I'm going to see any significant effect, but amazingly, like there is. So everything you see in gray are the control ones. So these are plants that have everything, like all the microorganisms on the surface are there. And the green box plots are the ones without all the microorganisms on the surface. And so the larvae, when there are no microorganisms, it's more. So like bad for the plant, right? Because it's being eaten more when it doesn't have like all these bacteria and all these microorganisms on the surface. But the larvae is less efficient, so that means that it has to eat a different amount of tissue in order to achieve a certain mass before pupation. And also, when the insect is already like an adult, they are smaller when they eat those leaves without the epiphytes. And so these can be like, there are several forms in which you can actually explain these results or make sense of these results. But it seems, or I like to propose that it seems that having that layer of microorganisms on the surface of your leaves is actually like the first line of defense whenever there's an herbivore present. And so it seems that understanding how these communities assemble and if it's stable or not to different perturbances might be important. So what we wanted to know is if this epiphytic community can actually represent the first line of defense, and if this is true, then how this community is assembled. And so Carmen did a series of experiments where first we wanted to know, like, so we already have the pairwise interactions, like the values of the interactions among like different bacteria. So we wanted to know if, based on those pairwise interactions, we can actually build like a synthetic community. Then we wanted to know like if that community was stable to the presence of a biotic disturbance, and also how the growth medium in which the community is growing is also like being affected or not by changing the medium every 2 hours or not. And so this is our synthetic community, and the reason we use these bacteria is because it's cheaper, and you can actually, like when you play them in a petri dish, you can actually see them growing different. So they have different colors, they have different texture, they have like different size of the colony. So I don't have to spend a lot of money, state university, so like I have to be clever about like how I do the experiments. So these are the first three ones are the two species that we used to first build the community and see if the community was stable. And then we use the Sporosarcina species with the green arrow to see if once this community is already stable, what happens when you introduce another one, right? And so first we wanted to model it, so using a generalized Berka-Volterra model, we first model if this community of 3 species was actually stable. So here you have so this is the result of the model, right? So you have here the relative abundance of each one of the bacteria we use. We started with equal abundances of the 3 species. And so here you can see that there's like a period of 4 hours where the community is stabilizing. After 4 hours, we introduce the 4th species, and then there's an unstable region, and approximately at 6 hours, the model tells you that the community reaches like some stability. And so this is what the model predicts, but then we wanted to know if this was actually true with experiments. So we have like all the possible combinations without refreshing the medium, with refreshing the medium, and without the presence of the 4th species, and with the presence of the 4th species. And so when you compare like the different, the relative abundances of all the possible combinations, you can actually see, I'm going to skip this. So if you estimate the symptoms diversity, here we have the ones that are with the perturbance and without refreshing the medium. So the resistance of this community to the perturbance is actually higher than the resistance of this community when you are refreshing the medium. And so that means, I mean, we don't really know what it means, but it means that whenever we're building a community that is not only with 2 species, but 3 species, like the relationships are changing. And that interaction, those ecological interactions are also changing depending on the medium they are growing. So the only conclusion actually I have is that I need to keep doing experiments, and I think like I have to keep like working with this community. And so what's next? So right now I have only told you something about like the epiphytic community and how it affects the performance of the insect, but we have like the other study, the other part of the history. So it's like how those bacteria from the gut of the insect affect like the expression of plant defenses. No? So we're working on that, and I also want to start working with sex selection because I know that there's like an effect on the size of the adult, and so I know that's it's probably important for how they mate and how they find a potential mate, etc. And just a reminder, Sue already told you, but I'm also part of the organizing committee for the second meeting of the Mexican something of biology and mathematics. It's in Querétaro, there's a QR if you need more information, so we hope to see you there. And thank you very much.
Hi, thank you for a nice talk. I was wondering if you created 2 networks at the beginning just using correlations and then by looking at cultures. So did you overlap them and did you have like the same result?
No.
No?
So we keep the 2 layers like separate. So just say that combining this, right, and just like overlay them.
Yeah, like comparing the results like from correlation and culture.
No, we haven't done that, but we can do it.
It would be interesting to see if you have the same results, if the correlation has power of prediction. My second question was, for the model, how did you choose the parameters for the...
These are the parameters for the local data model. They are from the experiments. So we know that the intrinsic growth rate of each species, and then we know that the capacity thing, yeah. So those are from the experiments.
From interactions you also measure.
Yes. Yes. Yes.
Nice talk. Thank you. I have a question about the 2 networks. You say that you have like the same structure, but you measure that structure?
No, I mean, if I say that they have the same structure, it was a lie. So actually, like I think we don't have the same structure, right? And the centrality is not the same. We have some other measures of the network, but I don't have them here. But I think essentially, although they share all the bacterial groups because of the nature of the interaction, like the structure is not the same. And we can only see that because we have like this multi-layer network approach, right? Yeah, but they're definitely not the same, yes.
Yeah, I have a
question.
Also, you say that you have positive correlation. I don't see it.
No. So the positive, I have a positive interaction. So when we did the experiments, or Anna did the experiment, some of the ecological interactions among the bacteria from the gut actually are positive, and I know that there's no way you can see, but here there are some like blue lines that show positive interactions. And that is with this formula. So if you have like a positive value, it's because you have like a positive interaction. If you have 0, it's that there's no effect of interaction and negative or yeah. So we only found, there are like 4 positive interactions, but only among the bacteria isolated from the gut. Thank you.
Go ahead, please. Well, about how a large fraction of the strains you saw in the metagenomic analysis could you also cultivate?
So we have several isolates but it's like less than 1% of everything like we were able to get from the sequencing analysis. But then the thing is that when we started isolating, like we only isolate those that look actually different, like in a petri dish. So there are more. So probably there are different species, but they look the same. And for the experiments, just for now, it's easier just to work with those that look different. Yeah.
I guess it's similar. I think that I'd like to go a little bit deeper into what are the species and not just the genus.
So
because, you know, like, I'm just interested in that. Why is Bacillus communis the bad guy? I mean, it's the same thing in my community, and it's the same thing in other places. And since I know the mechanism of action of these, I just wondered if I would just give you some of my strains to neutralize the bad guy, and we would get, you know, these working.
yes, please give me everything. Give me everything you have. Yes, I would love to. Yeah. Thank you.
I know that people really have very different biomes, and today it's used to help people digest better, etc., to make them leaner and what have you. How different are the bacteria of your, as someone made as a single insect has not differences between the microbiomes of individual insects?
So I know that, for example, if they eat leaves that are growing in the greenhouse, and you sequence like the gut microbiome, it's going to be different than if you collect the larvae right from the field. So, and also I think that, so we have several genetic families from the plant, and I know that they differ, for example, in defenses. I don't know if that can change like the microbiome of the plant. And so it could be really interesting, like if you have different genetic families of the plant and then you feed like the larvae and then sequence what's in their gut. So maybe you can see like differences also, like in the gut microbiome because of the genetic of the plant they're eating, right? But to be honest, like I don't know. I guess there are going to be differences, but I have no idea. The only thing I'm sure is that if you feed larvae from plants growing in the greenhouse, so their gut microbiome is going to be different if you compare that to larvae eating plants from the field. So I know that's the difference.